Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 4.1, cell communication. Today we're describing the ways that cells can communicate with one another, both through close and long distance, and we'll look at all the types of ways that this happens. And the suggested skill is Concept Explanation 1B, explain biological concepts and/or processes. So when talking about cellular communication and the first half of this unit, there's going to be a bit of a mantra that we're following, known as the signal transduction pathway, which consists of three parts: reception, transduction, and response. And these are going to make up cellular communication. So reception, there's going to be a chemical signal known as a ligand. Most of the time, that's the signal that's going to be sent out by the cell to be received by another cell, and it's going to be received by a receptor protein on the membrane of the target cell, or perhaps inside the cell depending on the ligand. And when the ligand enters the receptor, reactions are going to begin to transfer the signal to the nucleus or to the target that's going to create the response, such as, let's say, an enzyme that needs inhibiting. And this is going to be the signal transduction pathway, the transduction part of it. This is going to change the signal so that it is able to be amplified and create a response. And then obviously the response of the cell, the cellular response, is going to be what the whole end goal is of the signal transduction pathway. So before we get into the details of the pathway, we're going to talk about, just in general, different kinds of cellular communication that will then bring about a cellular response. The first two kinds of cellular communication that we're going to be talking about are going to be autocrine and juxtacrine signaling. So autocrine is going to be a chemical signal that affects the cell it was sent out from. So this is essentially self-reception, and it's going to help reinforce the role of the cell during development and plays a key role in metastasis. So over here you can see an autocrine signaling pathway where the ligand is sent out from the cell and then received by the cell itself. So what does this mean that it helps reinforce the role of the cell during development? Essentially, if the cell is producing the right chemical molecules and the right receptors, and it's sending them out to itself, then essentially that means that the cell is developing how it's supposed to be, and it's creating the right chemical molecules and the right receptors. Additionally, this is going to be part of the metastasis, which is when cancerous tumor spreads. Now, another kind is going to be juxtacrine signaling. These are signals that travel by cell-to-cell contact. So these are two cells, but they have to be touching in some way in order for the signal to be transferred. So some common ways this happens are through plasmodesmata, which are pores in the cell wall of plant cells, or gap junctions, which is sort of the same thing in the cell membranes of animal cells. And it allows intracellular mediators to diffuse between the cells, which is then going to create the transferring of the signal, and it can also create coordination between the cells because now they're going to be touching. And two cells can also bind due to complementary proteins, and one of the proteins is going to get changed. And this is often used by the immune cells to mark self cells. Essentially, the immune cells are going to mark body cells that are healthy, and then it knows not to then target them when it's fighting infection or whatever, because these cells are healthy and they're self cells. And additionally, in the immune system, helper T cells are trained by direct contact with antigen presenting cells that look like antigens. So then these helper T cells and killer T cells are able to kill infected cells with direct contact. So the entire immune system runs on this juxtacrine signaling, this cell-to-cell contact, because T cells are going to be trained by contact to know essentially what kind of antigens they're looking for, and then the killer T cells are going to be then killing the infected cells with direct contact. Number 3 on the list is paracrine signaling, and this is going to be a communication across a short distance. So the cells aren't touching, but it's going to be a local coordination of activity with neighboring organisms or cells. So one example of this is going to be synaptic signaling, essentially between nerve cells or neurons. And between neurons, there's going to be a release of a ligand known as a neurotransmitter. And when it's received, it can cause a change of the electric potential. So over here we have two neurons, and there's going to be a neurotransmitter transporter that's going to release the neurotransmitters, which are then going to be received by the receptor, and that's going to cause a change of electric potential, and then as the nerve signal is going to be carried across between neurons, allowing you to feel things. Now, then the ligand can be taken up by the first neuron in order to then send out the signal again once there's a new sensation. Now, this happens in single-cell organisms as well, and prokaryotes in the form of quorum sensing. And bacteria release autoinducers to determine population density of the species in a local area. So essentially, when the bacteria releases autoinducers and it receives them, and it can tell that there's a low concentration of autoinducers because not many are being recepted, it knows that it's alone, and it's going to execute behaviors that an individual bacteria would do. But if there's a high concentration of autoinducers that are then being received in this general area, this close area, then the bacteria knows it's not alone, and it's going to be performing in group behavior. So the gene expression and the behavior that the bacteria will perform is going to be different depending on the amount of bacteria in the area and the paracrine signaling. Lastly, morphogens are signaling molecules in embryonic development, and they influence the cell behavior of embryos based on their concentration in different areas. So paracrine signaling is especially useful for local coordination and the nervous system. Lastly, we have endocrine signaling, and this is going to be long-distance cell communication through the use of hormones. And they are going to be traveling through the bloodstream. That's how they're going to be able to cross these long distances. And some common examples include insulin, which is going to regulate glucose levels. When there's a lot of glucose, a lot of sugar in the body, insulin is released, and when it is recepted, it's going to promote storage of glucose as glycogen in the liver. And glucose will be then imported from the bloodstream, all because insulin was released from the pancreas and then received in the liver. Now, human growth hormone grows skeleton and cartilage and promotes cell division. There are thyroid hormones, and then lastly, of course, there are going to be estrogen and testosterone, which are going to be your hormones that create reproductive function and work throughout puberty, and they travel through your body through the endocrine system, through the bloodstream. So, cell communication review. Make sure you know the steps of the signal transduction pathway with reception, transduction, and response. And then, of course, the autocrine, self-receiving, juxtacrine, cell-cell contact, paracrine, nearby distance, and endocrine, long distance through the bloodstream signaling pathways. And we will continue talking about the signal transduction pathway in 4.2.